Did you guys worked on the homework question that I gave you about Max? What is the conclusion you came up with? Antivenom, yes, but what is it, that antivenom thing? Is it a specific medicine? Is it a vaccine? What is it? What do you think it could be? Antibody. So basically he needs to be treated with some kind of antibody with basically a serum, right? So which is rich in antibody. Now, so that is your answer, and the question is that if this is the case, then what kind of immunity he is actually receiving against that? So firstly we came up with the treatment. So he should be given with a serum which has some kind of antibody. That's right. Now question is then what kind of immunity he is developing? Is this an active immunity or a passive immunity? Passive immunity, because he's technically given sort of a medicine, right? So it is not active immunity. And if that is the case, anyone, do you think he will establish any immunological memory? No, because it's a passive immunity. It's just being treated. And why is it so important? Because this is a poison. So we cannot wait until his actual natural immunity kicks in. Like we cannot wait for 3 days so that he is given with some medicine, then the actual immunity will kick in. So that's why he has to be treated immediately. Now, anyone, if you have read through, what is the specific type of antibody you think he can be given? We have read about 5 classes. So what is the specific one that can be given to him, that can be applied towards toxins specifically? It can be IgM, because IgM can be given to any primary response. But there is a one that is specifically given for toxins. IgM might work or might not work. So we need something more specific. Go through the antibody classes and tell me. IgG, that is correct. So here IgM is the initial one that you can give to any sort of primary response, but not necessarily it will work in this case, because we need something fast and something direct. Therefore we need to have him IgG, which specifically works against toxins. So therefore, if we are giving him the serum or the supply of antibody, then it should be rich with IgG. Okay? Make sense, everyone? All right. So we will finish talking about antibodies. We'll talk about the antibody functions, what kind of functions antibodies do, and then we'll get into T cells. So first is neutralization. The first function, neutralization. This is the simplest function, which means that they kind of become defensive, the antibodies. So these antibodies simply prevent the antigen, whatever the antigen is, to bind to a tissue. Not necessarily they are attacking the antigen or marking it for destruction, simply neutralizing their function, reducing their function by preventing them to bind on a tissue. That is called neutralization, the simplest one. And when it will still itself bind to the antigen, and the antigen antibody complex will then undergo phagocytosis. The second one, which you should know about, is agglutination, which is antibodies can bind to the same determinant on 2 or different antigens. So here actually the antigen antibody complex form. And where have you read about this? Example, blood typing. So if there's a mismatch of blood, then we see this kind of reaction. So a person with A, if you give a blood group of B, then they will perceive each other as antigen antibody, and they will actually form agglutination, clumping. So that's a simple most example of that. The third one is precipitation. What is precipitation? Here also the antibody binds, but the soluble molecule is actually it is not binding to the soluble molecules actually form complexes instead of cells. So here now the antigen antibody binding, and then that whole complex of this molecule and then the antibody will undergo destruction. Okay, it's not attacking the entire cell, just the molecule. The fourth one, which is the most complex one, which actually also involves the complement trigger. So here the main antibody defends against cellular antigen, and several antibodies in this case can bound on the same antigen. They form an antigen antibody complex after that. So several antibodies can bind towards the same antigen. They form a complex, and after that it triggers the complement pathway activation. Okay, if the complement pathway is activated, then after that it will complement pathway can either trigger phagocytosis or it can trigger inflammation, any of that. So that will happen, and finally after phagocytosis or inflammation have happened through complements, then the cell will undergo cell lysis. Okay, the whole thing after it is being done, then whatever the product is created, that will be undergo cell lysis. So complement fixation and activation. So this is where the antibody is taking help from a complement. Now, other than bacteria, or we also can have parasitic infection. For example, worms like Ascaris, actually can also attack us. But worms are too big for these kind of reactions: precipitation, lysis, agglutination, neutralization. They are too big. So a small molecule like antibody will not be able, because it's a big organism, it will not be able to take care of that. In this case, two things will happen. IgE antibodies will still play a critical role. What really will take care of this, the eosinophils will actually bind to the exposed stem of the antibody, and this eosinophils itself will actually try to lyse the worm or actually release their toxic content. The eosinophil, therefore it is specifically designed to take care of parasitic infection. So the IgE antibody itself will not be able to do much, but it will take help from the eosinophil WBC. Eosinophil has its toxic content, toxic in the sense toxic for the worm, not for us, and then it will lyse the worm itself. So parasitic infections is taken care by eosinophils with the help of IgE. There are monoclonal antibodies. Now these are not natural, these are commercially prepared pure antibodies that is specific for one antigen, specific antigen. Okay, this produced by hybridomos, cell hybrids formed by fusion of tumor cell and B cell. This is mostly used in research purpose, clinical testing, cancer treatment. It is not generally, we don't produce monoclonal antibodies in our body. All right, question for you. The antibody shown, which antibody actually circulates in blood, is a potent agglutinating agent, which of this option is a potent agglutinating agent? IgM. Why do you say that? IgM is the right answer, but why do you say that? Quickly.
So that's what's listed on the description for the IgM?
Huh?
But the agglutinated agent is what's listed under the...
Yeah, so this is because this is the first immunoglobulin class, and this is the one that is required as a primary response, and there are numerous antigen binding sites that is present in here that will make it more suitable for becoming an agglutinating agent, right? So IgM. Second question, which of these phrases correctly describes passive immunity? Read the options carefully. Passive immunity. A. Actually, naturally acquired antibodies from mother to child. That's a passive immunity. Let's quickly recap that thing. So passive immunity, you can naturally get it like from your mother, or the infant can get it from mother's milk or from placenta. Or we discussed the case we just now discussed where we can actually have gamma globulin, which is serum, which will act sort of a medicine. So that will be the artificially acquired passive immunity. About active immunity, we can naturally acquire it when a virus or some pathogen or some bacteria will directly attack us, and then we will all develop the memory cells for it. Or we can get it via vaccines or attenuated or dead pathogens, and then we are exposed to that, and then we will learn the process to develop memory cells. Which one takes longer time, active or passive, to get like work on us? Which one do you think? Active. All right, so that kind of finishes about B cells. Now we'll talk about T cells. Okay, I'm going to split this into 2 lectures, T cells, because it is a slightly like a little bit complicated than B cell is. So this is more about cellular immunity. Okay, we are talking about process of activation and clonal selection of T cells here. There are different kinds of T cells, or they have different types of T cells that work a little bit differently like B cells. And then we'll finally talk about the T cells' functions in our body. Okay. So, questions? Anything? So this is now, so far we spoke about humoral response, okay, outside cell. Now we are talking about cellular immune response. T cells provide us defense against intracellular within cell antigen, protect against intracellular antigens. Okay. For example, any cell that has been infected by virus, bacteria, or a cancerous cell itself. So these are all intracellular cell or a foreign cell, transplanted cell, but something has to happen within. And there are 2 ways T cell can actually work. First of all, T cell also can develop memory cells, that is common part. But T cell either can directly kill the cell that it is like thinking as a problematic cell. It can directly kill that cell by releasing some chemicals, or it can actually mark it for destruction. But T cells are more complex than B cells in classification and function. Can B cell directly kill an antigen? No. What it will do? How will it work? It will not directly kill it. Then what will it do? It will mark it so that either T cells can do it, or maybe innate system can do it. Something it will not going to directly kill it. How will it mark? One of the way is antibodies. It can use that, right? So, or it can memorize also. But T cell is a little bit different than that. One type of T cell actually can directly kill the antigen. So there are 2 types of T cells, 2 populations based on the type of glycoprotein that they show on type of glycoprotein receptor they have. There are 2 types. Firstly, CD4 cells, and the second one is CD8 cells. CD4 cells can, firstly, both of them can produce memory cells. That is common, CD4, CD8. But CD4 can actually become helper T cells or TH cell. Remember, this is a helper T cell. So it helps other things to function. So therefore, it can activate B cells, it can activate other T cells, which is nothing but the CD8 cell, or can activate the macrophaging process, or can direct some other adaptive immune system. So it will help Again, it is sort of has a similarity with like B cell functioning. It will itself not kill the antigen. It will help other cells to do it. Or it can also become a regulatory T cell, which moderates the immune response. Kind of similar function, is just minute differences. So CD8 can become either a memory cell or it can become helper cell or regulatory cell. Now, I'm sorry, CD4. CD4 can either become a helper or regulatory cell and also can produce a memory cell. Now, how is it different from CD8? The CD8 is a cytotoxic T cell, which is the TC cell. TC cell. This cell, however, is really different from B cell or CD4. They are capable of directly destroying the foreign antigen. Okay, they don't mark it for destruction or anything, but they are directly capable of destroying the antigen. Okay, so therefore all these, including the TC cell, which is the cytotoxic cell, the regulatory T cell or the helper T cell, they are the activated cells, effector cells, which actually do the effect. Okay, but when they are not effective, when they're not doing anything, then we call them as naive T cell. And naive T cells are two categories, either CD4 or CD8. They have a same process of getting activated as B cells. So this is helper T cell. So we do know that T cells mature in which organ they mature? Thymus. They will be formed in red bone marrow. Now they will migrate to thymus, right, where they are maturing. And maturing means they are developing their receptors and everything. So, and they will mature into two types, CD4 or CD8. So they are matured in thymus and they will stay there as CD4 and CD8. Now up to here, CD4 or CD8, we can call them as the naive T cell. They are not yet activated. They're simply sitting there. They have matured. What is the process of activation of lymphocytes in general? How are any lymphocytes, B, T, anything, how are they activated? When will you call them that this lymphocyte is activated now?
When they interact with the pathogen.
Exactly. Before that, they are matured, but until they are actually interacting with a pathogen, which happens in where? Part of the lymphatic system? Lymph nodes, absolutely. So when they interact with the pathogens in the lymph nodes, now they are activated. So the same thing happens for T cells also. Now they are activated. Now when it comes to T cell, the antigen that we are talking about, they can be presented by an APC or cannot be. Let's say for now that the antigen is, which is dendritic cell, that is presenting it the antigen. Okay, APC is there, and after that it is activated. Now the APC, antigen presenting cell, will have specific type of MHC molecule displaying on them, class 1 or class 2, which you can see here, which we will talk about just in a second. But remember this, that APCs, or the dendritic cells in this case mostly, will have two kind of proteins will sit on them that can display the antigen, which is MHC2, MHC1. So it simply means for now that the T cell is now activated because it is being presented with the antigen. So after it gets activated, what happens now? Firstly, both can form memory cells, CD4 or CD8. Okay? A part of these activated cells will form the memory cells. Will form the memory cells, and that is common for CD4, CD8. Now they are proliferating, so the rest of them will form, actually, if it is a CD4 cell, then it will form T helper cell or T regulatory cell. If it's a CD4, then it will form the T helper cell or T regulatory cell. But if it's a CD8 cell, then it will form a cytotoxic cell. Okay, if it's a CD8, then it will form the cytotoxic cell. So after maturing, after activating, when it is maturing the T cell either into CD4 or CD8. Now it is activated because it is now being presented to the antigens. Okay, now after it is activated, now it will start proliferating. Part of them will become, doesn't matter CD4, CD8, part of them will form memory cell, that is common. Okay, but if it's a CD4 cell, then they will develop or proliferate into T helper or T regulatory cell. If it's CD8 cell, then it will form the cytotoxic T cells. All of these cells, helper, regulatory, or the cytotoxic cells, these are all effector cells. Okay, these are all effector cells, means they are the one who will actually create the effect. We saw the same thing in B cell also. B cell also will proliferate into effector cell and then memory cell. Effector cell also called as plasma cell, which produces a special type of molecule. What is that? What? Antibodies. So in B cells, those plasma cells or these effector cells produce antibodies. Now these effector cells, which are 2 types or 3 types, they will have different kind of function. But here we have 3 types of effector cells, either TC cells or T regulatory or T helper cells. Alright, I'll give you a little bit break, a minute, then we will get into the next step. Overview this figure really well. This is a super important one. After you get the basics, then we'll get into MHC too. MHC. Understand what is happening here step by step. All right, questions? Any doubts, questions? So is the thing clear? Couple of things you have to remember. What is naive cell? What are the 2 naive type of T cells? What are they maturing into? What happens when they are activated, and what does it mean by activation, and what happens when it proliferates? Okay, and then after it proliferates, what does it become? What are the effector cells and then the memory cells? Right, so these are the stuff. Now now I told you, whenever a T cell is activated, that is true for B cells also, their only time they are activated when they are interacting with an antigen. Now T cell has a speciality in this case. T cell will only respond if the antigen is displayed on surfaces on cells by MHC protein. So a cell have to have an MHC protein and the antigen has to sit on that. Then only the T cell will recognize that. So remember that. So T cells only responds to process fragments on the antigen or the antigen itself that is displayed on the surface of cells by major histocompatibility complex or MHC proteins. So there is, suppose a T cell, it will recognize an antigen, but how will it recognize? Provided that antigen is presented to the T cell on an MHC protein. So the antigen itself has to sit on an MHC protein, and this whole complex of MHC protein and the antigen, which is this thing, has to sit on an antigen presenting cell, or can be other cells also, but antigen presenting cell or some cells. So the cell will have the MHC protein, and then it will have to have the antigen attached to it. Then only the T cell will recognize that antigen. Does it make sense? Everyone, make sense? If the antigen, in other words, is not presented on an MHC protein, T cell will not be able to recognize it. Okay? Now, antigen presentation is vital for the naive T cell so that it can become on effector T cell. And when I say vital, that means it has to be presented on the MHC protein. Otherwise, the naive T cell is not going to recognize it. And there are two kinds of MHC proteins, class 1, class 2. So class 1 MHC protein can be present on the surface of any cell other than RBC. Okay? Can be any cell other than RBC, class 1 can sit on that. But class 2 is only present on the antigen presenting cells, like dendritic cells, macrophages, or B cells. These are the three antigen presenting cells. So class 2 will be only on antigen presenting cells. So this means that antigen presenting cells will specially have class 2, but it can also have class 1. Right? Because class 1 can be present in any cells other than RBC. And RBC is not an antigen presenting cell. So therefore, every cell, most cells, will have class 1 MHC proteins other than RBC. And this also includes APC. Now, APC will specially have another protein, which is the class 2. Alright? Because it is an antigen presenting cell, so it is given an additional tool, which is the class 2 MHC protein, to present the antigen to T cell. Is it clear? Alright. And all these MHC proteins are synthesized in ER, like any protein in our body. Now what happens? So this is actually the crucial part in this chapter. So class 1 MHC protein. So first we'll talk about them. Which cells can display class 1 quickly? Can APCs do this? Antigen presenting? Yes, they can. So class 1 MHC protein binds with shorter antigens, 8 to 9 amino acids, and they are endogenous antigen, means they are either abnormal proteins within the cell or maybe self antigen, normal proteins of the cell. So simply short antigens, endogenous antigens. And this is crucial for CD8 activation. So see here what happens. So suppose all nucleated cells, obviously the cell has to have a nucleus to this to happen. So this is the cell, and it will have class 1 MHC present in that. Okay, here cell with class 1 MHC, and then the antigen will sit on that. Okay, the antigen will sit on that. This whole complex, when this whole complex is formed, then only the CD8 will be able to recognize. Okay, CD8 will be able to recognize this complex, provided that the cell has a class 1 MHC and then the antigen is sitting on that. Okay, and the antigen will be an endogenous type of antigen. After this, actually the CD8 is directly going to kill the cell. Okay, it will have the cytotoxic, it is a cytotoxic cell, right? So it will be able to directly kill the cell. We'll come back to this. For example, here, this is the whole cell with the antigen and then the MHC1 class 1 protein, and this is now being presented to this is the complex, the cell with MHC and then the antigen. Now this is being presented to the CD8 T cell, and what will the CD8 will do when it interacts is directly going to kill the infected host cell. So it will mark this, that whatever it is, this is something foreign, it doesn't look good to me, it is being presented to me by a class 1 MHC, so I'm going to kill it. That's it. It will directly kill that. Alright. Now what happens to the class 2? Now class 2 has a little bit specialized requirement, because now not any cell will be able to present. We need the APCs for this. But this actually works with an exogenous or slightly longer antigens, and they can only sit on the antigen presenting cell. So in this case, the APCs actually, the dendritic cells here, they are the one who has the class 2 MHC, and now they're added with the antigen. So they have formed the complex, the class 2 MHC and antigen. Only APCs will have class 2. Now they are being presented, but they are presented to CD4 cells, not CD8 here. So they are now presented to CD4 cells, not CD8. So therefore, and this is an exogenous, a longer antigen. Now in this case, it will not directly going to kill, but it is going to mark it for destruction, for example, like phagocytosis or some other form of destruction. It will take help from another type of destruction. It will not directly kill the antigen. Okay? So it will going to create the defense system. So therefore, CD4 and CD8 have different requirements of MHC. So CD8 is only works with class 1, CD4 works with class 2, right? CD8 works with class 1 and CD4 works with class 2 of MHC. Now CD4 cells become helper cells, T helper cells, and restricted to bound only class 2, and class 2 is only present on antigen presenting cells. Class 2 MHC will not be present on any other cell. Now the CD8 is different. It is a cytotoxic T8 cell. Now it will become the cytotoxic T8 cell, which is the effector cell, but they will be restricted only to class 1, and this class 1 MHC protein can be presented on any cell including APC, but the only exception here is RBC. Okay? Now once activated, the cytotoxic T cell will seek out the same antigen and basically it will destroy that cell or destroy the antigen. So on class 2 here, when it is presented with by the dendritic cells or the APC, it will have class 2 MHC as well, will have the antigen present on it. So this is the whole complex, and then that will be presented to the CD4, so whole complex presented to CD4, and then it will mark it for destruction. It's not going to kill like CD8, but it will mark it for destruction. It will create a pathway. It can be complement depending on the antigen. It can be a complement, phagocytosis, inflammation, anything. All right. So depending on this, think about it a little bit and tell me which cells, which lymphocyte cells can be responsible for tissue graft rejection. Think about what we just discussed about. It has to be a lymphocyte. That's the clue. But which can actually reject or destroy that whole tissue graft? Rejection means it will destroy, it will not let it survive. CD8, which is the TC cell. Very good. The cytotoxic T cells or the TC cell will actually can do it. Rejection means it will actually not even accept it. It will try to destroy that. Tissue graft rejection means that. Okay. So if you have a tissue graft rejected, it's not matching, it will just mark it for destruction, try to destroy it. Quickly a recap, what is antibodies mode of action? Slightly different from what TC cells do. How antibody works? Come on, you know this. How antibody works? Does it kill the antigen directly? No. Then what does it do? It immobilizes, inactivates it, tags it, and marks it for destruction, which is very different from TC cells. CD4 rather will kind of do the similar thing, but not the TC cells. Okay, I'm going to stop here, and then this again for next class we will finish T cells, and I purposefully actually covered this short. Before you come to next class, make sure you go through this lecture again, okay? And then because the next part is also not very easy, okay?